Hello, this is Mrs. Hawk, and I'd like to talk to you a little bit about the basics of viruses as to how viruses infect the cell and what their structure is. So, first, go find the sheet that looks like this, and if you want to print it out, you can print it out and then you can follow along. But let me reveal for you, first of all, the basic virus structure. And this looks to be some sort of a coronavirus, corona meaning crown. And the idea is that if a virus attacks onto a host, and they're very specific to which species they can attach onto because of the parts of the cell that the host will have. And the basic steps are to attach onto the cell's surface. So that's going to fit in this particular blank right there in that square. And then the cell takes the virus into it, and once it's attached on, then the parts of the virus will go into the virus or into the cell, and the virus will start assembling new cells. It's not likely to attach onto a different organism because it doesn't have the right markers on the outside of the cell. We'll talk a little bit more about that later. And what eventually stops it is this is going to be the immune system going in and getting rid of the virus. And the DNA of the virus is going to, what occurs after the DNA is released? Into the host cell. Once that DNA is in the host cell, then it can start assembling, the cell can start assembling the new little virus pieces. So I really like to think of the virus as a pirate that goes in, puts its own directions here in the form of DNA into whichever cell it's attaching onto, and then the cell that it attaches onto starts making little virus parts. So the first thing is to take a look over here on the second page, and this would be a good page to go color yourselves. But this outside is this looks a lot like a cell membrane, and it is a couple layers thick, and it asks us to color it yellow here. This is a 2 layers thick. It's a phospholipid bilayer. And the phospholipid bilayer is fat on the outside, or water on the outside, water-loving molecule on the outside, water-loving on the outside, fatty on the inside. Lipid here, and then the inside of the cell, or inside of the membrane, is water-loving. So this part right here is really a blow up of what this looks like. And we're going to talk a little bit later about how those lipid pieces and water-loving pieces, we can get those to combine together. This is a 2-layer thick layer of envelope around the outside. And the proteins attached onto the outside, we're going to color those red, and it's labeled B. The proteins right here. These are, a better word to use here is the word antigen. And an antigen is a marker that's on the outside of a cell. And the markers on the outside of every cell, or I guess in this case of the virus, are going to be very, very specific to what that virus looks like and what it is going to be able to attach onto. It acts like a little lock, and it tries to attach itself onto the exact cell that it is designed for. And the reason that viruses don't typically go between different species is because these antigens, these markers, will be very specific to whatever it is hosting. And the capsid then is green, which is here. Ooh, that's a pretty green. And this is now kind of like a nuclear membrane of a cell. And what it does is it adds another little layer of protection around the information that's on the inside. And then the DNA here on the inside is blue. Now, the great thing about being a DNA structure is that DNA is double-sided. So that means that there's a spell checker to the DNA. If you remember, DNA, you have a C on this side and a G on this side, and those will always connect with each other. An A on this side will always connect with a T on the other side. A T on this side will always connect with an A on this side, and a G over here will always connect with a C. So what this double layer of DNA, or double strand of DNA, allows is for this side to remain the spell checker for this side, and vice versa. There's always somebody checking to make sure we're correct. And then the last part is here on this inside, there is tegument. And we're going to color this purple. This is the fluid. It's got proteins. It's got some things that are going to be able to help the virus to do the things that it needs to do. And this is the basic structure of a virus. This happens to be a DNA virus. We're going to take a look at the next one at how the specific virus of HIV works.